Everybody, Dr. Becker, Chapter 3, Molecular Biology, Genetics, and Genetic Disorders. Another review and blast from the past for us to review this kind of content. In Chapter 3, they're really going to focus on genetic disorders and how diseases are inherited and some aren't. We have to understand how genes and the genetic information is passed down through generations. We have to understand how mutations work and how gene activity may be modified, and then kind of talk about the future of research and those things as well. Key concepts are listed here. This also comes at the end of your chapter, and I will let you guys read through that a little bit later. So these are some of the disorders that can be genetic, are listed here. We did talk about phenylketonuria already a little bit as well. So the summary of cellular information transfer, okay? There are two primary roles for the DNA and RNA in our cells. On a minute-to-minute basis, our cells will synthesize proteins that are based on the processes of gene transcription and messenger RNA, which we call mRNA translation. So these proteins are often unique to the cell type, and they are tightly linked to structure and function of the differentiated cells. So on one hand, cells age and must be replaced with new cells that are specialized to take up the structure and function of their precursors. Also, generations of descendants require that cells be duplicated many times over in the process of sexual reproduction, fertilization, embryogenesis, those things. Meiosis produces gamete germ cells that are haploid, etc. In fertilization, two haploid germ cells will combine and they'll produce the normal diploid cell with two copies of each chromosome. Okay, so we have this method of reproduction which adds to overall genetic diversity by bringing together information from two genetically different parents. The DNA contains the cell's genetic information in a very stable, double-stranded molecule that can be faithfully replicated and used to produce daughter cells with the identical information present in the parent cells. So DNA also provides the template for transcription of RNA needed for new protein synthesis by translation. What does all that mean? This is figure 3.1 here in this slide, and this shows the nucleic acid functions. So the role of nucleic acids in the transmission of genetic information and instructing protein synthesis is kind of outlined here. So we can see DNA is replicated prior to cell division, providing daughter cells with identical genetic information. And the genes found in DNA are the instructions transcribed into RNA and then are used as templates for translation of proteins. At various steps in the process of germ cell formation and cell reproduction, changes in DNA sequence can occur. When those variations, or what we call mutations, occur, cell programs often detect and correct them. So we have our own kind of police detective system within our DNA that notice, oh, that replication did not go well. We need to kill it off or not allow it to keep going. And so we have some checkpoints for that. But over time, some mutations turned out to be beneficial in some environments and were retained. So let's jump into osmosis and review generally DNA structure and some of these components before we go too much further into this lecture.
Information.
Start it over.
Buried deep within the nucleus lies our genetic information, called DNA, which stands for deoxyribonucleic acid. DNA is made of two strands that are coiled around each other in a double helix. Each of the two strands that make up DNA is a polynucleotide chain, so it's a string of nucleotides one after another. Nucleotides are organic molecules that are made up of a 5-carbon sugar, a phosphate group, and a nitrogenous base, also called a nucleobase or just a base. For DNA, the 5-carbon sugar is deoxyribose. Deoxyribose looks like a pentagon, and the tips of the pentagon are 4 carbons and an oxygen molecule. The 5th carbon is outside the ring, and it binds to a phosphate group. The sugar and phosphate elements are the same for the 4 nucleotides that make up DNA. The difference comes from the nucleobase, which is attached to the first carbon of the sugar. There are 4 nucleobases that make up and give DNA nucleotides their name: adenine, or A, thymine, or T, cytosine, or C, and guanine, or G. Structurally, these bases can be either purines or pyrimidines. The purines, guanine and adenine, are made up of 2 heterocyclic rings. The pyrimidines, cytosine and thymine, are made up of a single ring. You can remember this with cut pi, because cytosine and thymine, along with uracil, which is a nucleotide found in RNA, are all pyrimidines. The nucleotides bind to one another using their sugar and phosphate groups. The phosphate group on the 5th carbon of the sugar of one nucleotide, also called the 5' carbon, forms a covalent bond with the 3rd carbon on the sugar of the next nucleotide, also called the 3' carbon. This gives each DNA strand a sugar-phosphate backbone, as well as a direction. One of the strands runs from the 5' end toward the 3' end, while the other one runs from the 3' to 5' end. This makes DNA an anti-parallel molecule. It's kind of like 2 snakes coiled up together but facing different directions. To form a double helix, though, the nucleotides use their bases, A, T, C, and G, to form hydrogen bonds with bases on the opposing strand. Bases form bonds according to the rule of complementary base pairing, which states that in DNA, A always pairs with T by means of 2 hydrogen bonds, while C always pairs with G through 3 hydrogen bonds. The hydrogen bonds are a lot weaker than the covalent bonds that hold the strands together, so they can be easily broken and then reformed when DNA is being transcribed into RNA, or being replicated during cell division. Now, DNA is actually a super organized molecule, because the 2 strands coil around each other once every 10 base pairs. This twisting and turning makes the DNA molecule develop major and minor grooves, which are larger or smaller spaces between the strands where proteins can bind to DNA in order to regulate its functions. DNA is also a surprisingly long molecule, over 2 meters long when fully stretched. To make 46 of these DNA molecules, meaning 1 molecule for each chromosome, fit into a tiny nucleus, our cells rely on a few packaging tricks. The first step is to take the double-stranded molecule and wrap it around special proteins called histones. Histones come in groups of 8, arranged in 4 stacks of 2, and they're positively charged. Meanwhile, the phosphate groups on the DNA strands are negatively charged. These opposite charges attract, so the double helix wraps twice around each group of 8 histones, forming a nucleosome. Nucleosomes look a bit like beads on a string, and they can snuggle close together and be further packaged as chromatin fibers. Chromatin comes in 2 flavors, euchromatin and heterochromatin. With euchromatin, nucleosomes are more loosely packed, because it contains the genes or regions of DNA that the cell frequently uses to make proteins that help the cell perform its day-to-day job. With heterochromatin, on the other hand, nucleosomes are more densely packed, because it contains genes that the cell rarely or never uses. Throughout most of the cell's life, chromatin fibers float in a loose state inside the nucleus, like DNA-rich noodles. However, when the cell is preparing for cellular division, the DNA noodles become more organized so that the DNA can be replicated, and each of the 2 resulting daughter cells can get an identical copy of the genetic material. Before replication, each of the chromatin noodles represents a single chromosome, which is made up of a single chromatid that contains a single DNA molecule. So 46 chromosomes, 46 chromatids, 46 DNA molecules. During replication, each chromatid is copied and pasted, so the amount of DNA doubles up. The 2 resulting chromatids are identical to each other and to the original genetic template, and they join together in the center in a region called the centromere, but they still make up a single chromosome. So following replication, even though there are still 46 chromosomes inside the nucleus, they're made up of 2 chromatids each, so 92 chromatids, which correspond to 92 DNA molecules. After the cell divides, the sister chromatids separate, and each resulting daughter cell gets 1 chromatid made up of a single DNA molecule. So back to 46 chromosomes, 46 chromatids, and 46 molecules of DNA, which once again look like chromatin noodles inside the nucleus. Alright, as a quick recap. DNA is made up of 2 strands that coil one around the other to form a double helix. And you cut pi, so the pyrimidines are cytosine, uracil, which is only relevant for RNA, and thymine, and the purines are adenine and guanine. Nucleotides have sugar and phosphate groups which form the sugar-phosphate backbone of DNA. Nucleotides on opposite strands form hydrogen bonds to keep 2 strands together. A binds with T, and C binds with G. DNA wraps around histones to form nucleosomes. Then the nucleosomes condense to form chromatin fibers. And finally, chromatin condenses to form 46 chromosomes.
That was helpful to kind of tie in where we ended in chapter 2 into where we are now in chapter 3. and talking about some of these things. So, cellular information transfer. All right, keep moving on. So again, the DNA structure is a very stable double helix held together by hydrogen bonds between base pairs. The backbone consists of deoxyribose sugars held together by the phosphate bonds, and the DNA bases and pairs. The A and the T go together and because of 2 hydrogen bonds, and the C and the G go together because of 3 hydrogen bonds. The backbones have a polarity based on the free ends of the chain, one of which is a phosphate attached to the 5' carbon of the ribose sugar, and the other is the hydroxyl attached to the 3' carbon of the ribose sugar. So the double-stranded DNA strands are anti-parallel to each other, with the 5' end of one strand opposite to the 3' end of the complementary strand. Okay, so when properly aligned, the adenine makes 2 hydrogen bonds with its complementary base thymine, while cytosine makes 3 with guanine. The comparative weakness of these hydrogen bonds allows the structure of the DNA to separate and rejoin with relative ease during the replication process, because we do this over and over and over again. Based on the complementary base pairing, the strand twists during formation, assuming the famous double helical structure. So again, in human cells, the DNA is found in 46 chromosomes, 2 copies each of 22 autosomes, which is 1 through 22, and then we have the 2 sex chromosomes, so females are XX and males are XY. The more than 3 billion base pairs of DNA distributed in the chromosomes range from the longest, almost 250 million base pairs, to the shortest, which is 54 million base pairs. Mitochondria have their own DNA also. The long strands could be subject to tangling and breaking if not organized in this fashion. So the first level of DNA organization involves wrapping around small protein bases called the histones. The histones serve as an anchor around which the DNA can coil, but are also active structures in DNA replication, transcription, and translation. So the histones are very important. As histones wrap around the DNA, the entire structure further coils. So prior to mitosis, the chromosomes appear in their most tightly packaged form. The 46 chromosomes of each individual form that individual's genome. So among the approximately 20 to 25,000 genes in each person's genome, there are large sections that contain non-coding regions. Those regions lack protein encoding genes and were originally thought to be areas of quote-unquote junk DNA. But ongoing studies find that these regions can impact gene expression or RNA and protein stability. So they're very important, we are now learning. Although there is considerable standardization of the base sequence across the human population, there's also variability in the genome. So the differences can account for our uniqueness as individuals. We share 99% of our genes with one another, but we also share 98% with chimpanzees, 92% with mice, 90% with cats, 85% with zebrafish, and 84% with dogs. I love that statistic. All right, so then we get into, sorry, I was recording on the wrong screen. Again, before mitosis, we have chromosome condensation. Each chromosome has 2 sister chromatids. The P arm is the short portion and the Q arm is the long portion. At each end, the telomere gets shorter with each round of replication. Eventually, replicative senescence is reached due to inadequate telomere length, so they kind of fall off and die. All right, DNA occurs, DNA replication will occur during the cell cycle. Now, the cell cycle is something I'm going to stop here and we're going to watch again on osmosis. Cell cycle becomes important in a lot of medications, especially for things like cancer and neoplasm and oncology care, because some of our drugs are meant to interrupt certain parts of the cell cycle. And again, for many of us, it's probably been a hot minute since we've learned about the cell cycle, so let's have a quick refresher on our osmosis here.
Cellular division. The cell cycle refers to the events that somatic cells, which includes all of the cells in our bodies except the reproductive cells, go through from the moment they're formed until the moment they divide into two identical daughter cells. This cycle varies in length depending on the type of cell. For rapidly dividing cells, like skin cells, it takes less than a day, whereas for other cells, like liver cells, the cell cycle can last years. The cell cycle has two phases, interphase and mitosis. Interphase is the longest part of the cell cycle, and it's a state of preparation, during which the cell carries out its cell functions, and grows and replicates its DNA to prepare for mitosis, or cellular division. After a parent cell divides, each of the two daughter cells enter interphase again. Now, interphase can be broken down into three subphases, G1, S, and G2. G1 stands for gap, or growth 1, and it's the longest phase of the cell cycle. During G1, the cell mostly grows, and the organelles take care of regular cellular business, like the synthesizing of proteins and producing energy. Inside the cell nucleus, there's our DNA, organized as chromosomes. And during G1, each chromosome is made up of a single, thin spaghetti of DNA, called a chromatid. At the end of G1, there's a cell cycle control point, called the G1 checkpoint, where the cell checks to see if the DNA is not damaged and it synthesized the right proteins in the correct amount. If it turns out that there is any reason for the cell not to divide, like having DNA damage, things can go one of two ways. The cell can either enter a non-dividing state, called the G0 phase, where the DNA repair mechanisms try to fix the problem, or the cell can self-destruct in a process called apoptosis. Now, if the cell does get the go-ahead at the G1 checkpoint, it enters the S phase. S stands for synthesis, because during this phase, DNA is replicated, so that each daughter cell receives identical copies of the genetic material. So for each chromosome from G1, an identical copy is created. This happens with the help of a number of proteins, both structural proteins and enzymes, as well as energy. Now, just to be clear, this doesn't mean that the number of chromosomes increases. Human somatic cells have 46 chromosomes throughout the cell cycle. The amount of DNA they have, though, and in turn their aspect, changes throughout the cell cycle. So each chromosome enters the S phase with a single copy of the genetic information, called a chromatid. During replication, each chromatid is copied and pasted, so the amount of DNA doubles up. The two resulting chromatids are identical to each other and to the original genetic template, and they join together in the center in a region called the centromere. But they still make up a single chromosome. So while the amount of genetic information is doubled, there are still 46 chromosomes that contain the genetic information. The cell can now enter the G2 phase. G2 stands for gap, or growth 2. Even after synthesizing copies of the DNA, the cell still has to duplicate organelles so that there are enough for both daughter cells. In fact, by the end of G2, the cell looks like a big balloon of cytoplasm and organelles, just waiting to split. Before it can do that, though, it has to first pass the final G2 checkpoint. And if there's no DNA damage after replication, the cell can enter mitosis. Now, mitosis is when it actually divides into two daughter cells, and it involves separating the replicated DNA in two distinct nuclei, a process called karyokinesis, and the actual separation of the daughter cells into two distinct cells, a process called cytokinesis. Mitosis can also be broken down into 4 subphases. In order, these are the prophase, the metaphase, the anaphase, and the telophase, and they're immediately followed by cytokinesis. You can remember this with the mnemonic, please make another 2 cells. During prophase, the membrane around the nucleus disintegrates, and the chromosomes condense and become visible under a microscope, like 46 little X shapes. The condensed sister chromatids make even the tiny Y chromosome in a male look like an X. Inside the cytoplasm, 2 organelles called the centrosomes migrate to opposite poles of the cell. During metaphase, chromosomes move toward the middle of the cell, on a line called the metaphase plate. So think M for metaphase and M for middle. When the chromosomes are in place, each centrosome sends out thread-like projections called spindle fibers that attach to the centromere of each chromosome. Next, during anaphase, the centrosomes start pulling on the spindle fibers to pull the sister chromatids apart, like a game of tug of war. Since both centrosomes pull with equal force in opposite directions, eventually the sister chromatids separate and reach the opposite poles of the cell. Finally, there's the telophase, during which a nuclear membrane forms around each new set of 46 single chromatid chromosomes. After that, during cytokinesis, the cell membrane pinches in until the 2 daughter cells separate. Now, both those daughter cells can enter the cell cycle once more if they need to. If they don't, they enter the non-dividing or G0 phase. G0 is considered to be a phase outside the cell cycle, because while the cells are living, they're neither dividing nor preparing to divide. An example of this would be hepatocytes, which rest in G0 until there's liver damage, at which point they enter G1 again, so that they can divide and replace damaged tissue. Another example would be neurons, which spend their entire life in G0, All right, as a quick recap, the cell cycle can be divided into two phases, interphase and mitosis. Interphase comprises of the G1 phase, during which the cell grows and performs its cell functions, the S phase, during which DNA is replicated, and the G2 phase, during which the cell grows again before entering mitosis. Mitosis can be broken down into prophase, metaphase, anaphase, and telophase, during which the replicated DNA divides equally for the two daughter cells, and ends with cytokinesis, which is when the cell membrane actually divides to form the two new cells.
That was helpful to have that quick review. And again, the links for these osmosises are on here, so you can go back through and listen if you need to, slower and however you need to review. So the cell cycle, again, let's go over it a little bit in more detail. The first three stages are collectively referred to as interphase and include the G1 and the S or synthesis phase and the G2 phases. During the G1 phase, the newly divided cells grow. So G1 is growth, synthesizes additional organelles, and engages in whatever the function of that cell might be. Some cell types have limited or no ability to divide once they have differentiated. They are then considered to be in the G0 state. The G1 checkpoint is a point of delay until appropriate signals indicate that the cell can proceed. So with those signals, including availability of needed DNA precursors and the needed synthetic enzymes, at the G1 checkpoint, it will permit the cells to enter the S phase. During the S phase, all the DNA is copied in the process of replication. So after the DNA replication, the cell enters the G2 phase. During the G2 phase, the cell continues to grow and perform its essential function as it also prepares for cellular division or the mitotic M phase. The G2 checkpoint then ensures that the cell's DNA has replicated correctly and enzymatically fixes any errors in replication that could have occurred. When the verification and repair process is successfully completed, then the cells can proceed to the M phase, which is mitosis. If the DNA has become too damaged and cannot be repaired, the cell is stimulated to undergo apoptosis, cellular death, and is destroyed. When we talk about the steps of mitosis, in the initial condition, we have a single diploid cell, which has the 46 chromosomes, two copies of each, and the DNA is dispersed within the nucleus. After successfully passing the checkpoints, each chromosome undergoes replication. Mitosis phase is prophase, metaphase, anaphase, all those that we aren't going to get caught up in. But during the mitosis phase, the nuclear membrane dissolves. The chromosome pairs line up along the midline. Those spindle fibers will pull the chromosomes towards the opposite ends of the cell. And then we go into telophase. The nuclear membranes will then reform and enclose the chromosomes, and the DNA disperses. Cytokinesis, plasma membrane investigates and completes the separation of the two now daughter cells, with each having their 46 chromosomes. And again, when we kind of go back up into here, it's a good example of those parts as well. I don't want us to get caught up in exactly all the things that I don't need you to memorize interphase and prophase or anything like that. I need you to know the general idea of what's happening. Okay. Now, steps in meiosis. To generate the haploid germ cells, which are egg and sperm, one round of replication is followed by two divisions. Recombination occurs after replication, increasing genetic diversity in the 4 end cells. So you have two daughter cells then are generated. So we have meiosis 1 and meiosis 2, and they have the same stages each as the mitosis. So meiosis also has prophase, metaphase, anaphase, telophase. In males, the progression through both stages is continuous, and spermatogenesis occurs from early puberty through late adulthood. But in females, meiosis begins during the first 6 months of fetal development and is interrupted at prophase of meiosis 1. At the time of ovulation, each oocyte completes meiosis 1 and again stops during prophase of meiosis 2. Then, when an oocyte is fertilized, that cell rapidly completes meiosis 2. The process of the meiosis is not completed in oocytes that fail to be released during ovulation or those that are not fertilized. So meiosis is very specific now to our sexual reproduction steps as well. There's a box 3.1 in your text that goes over the steps of meiosis as well that can help reiterate what we've just talked about. All right, recombination. So recombination of the portions of the homologous chromosomes can occur before the first meiotic division. So this image here is the exchanged by recombination between the sister chromatids. So the homologous chromosomes line up before the first meiotic division, and portions of the paternal chromosomes can exchange with a portion of the maternal chromosome. So we have this crossover. The egg and sperm have just one of each autosome and one sex chromosome. So genetic variability is promoted by mixing of maternal and paternal chromosomes. So that's a nice explanation of how, when mom and dad get together, how they're able to get their egg and sperm and how that exchange happens and we get a new combination. All right, DNA replication. The replication of the DNA takes place during the S phase of the cell cycle, and there is no difference in the replication process in somatic cells or gametes. The ability of the DNA to replicate depends on the unwinding of that double helix and the presence of the raw materials to make new strands. So we need free nucleotides. The activity of the enzymes that construct the new strands of DNA. Replication does not occur in a linear fashion, but rather occurs simultaneously at multiple points along the DNA molecule. So this simultaneous replication process permits the strand to replicate very rapidly. And since we have billions of base pairs that need to be lined up and joined, it's absolutely necessary to happen very quickly. So this multi-site replication also requires the connecting or ligation of the DNA segments, as well as the action of the topoisomerases, which work to prevent supercoiling of the DNA. So we kind of talked about this. At the either end of the backbone of a DNA molecule, there's a phosphate bonded to the deoxyribose 5' carbon on one end, and a hydroxyl bonded to the deoxyribose 3' carbon on the other end. So on the opposite strands, the ends are reversed. So in essence, the two strands of a DNA run in opposite directions, referred to as anti-parallel orientation. So by convention, the 5' end of the upper strand is presented on the left, with the complementary 3' end of the lower strand on the left. Okay. And they are able to be arranged in an orientation that will help the DNA to be replicated. The RNA primer initiates the copy strand that is elongated by the DNA polymerase. The leading strand can be synthesized continuously, while the lagging strand must be synthesized in segments as the replication fork moves to adjacent segments. Okay. That's a lot of things to know, but essentially you need to understand the 5', the 3', that they line up kind of anti-parallel to each other, and that's how the helix is formed. So in order to replicate, that process starts with the DNA helix unwinding, and as the DNA unwinds, the downstream portion of the molecules is subject to supercoiling, which is bad, right? And that will halt the process. But the DNA topoisomerases have the task of cutting the DNA molecule at very precise locations, and then that allows the coils to unwind by passing through each other and then reattach, and the replication can go safely and smoothly. Okay. This one here is an illustration of proofreading. So proofreading during DNA replication is able to identify mismatched bases and remove them before moving further down the strand. So this process of proofreading greatly reduces spontaneous mutation formation, which is really important. So if they find an error, they can fix it before it's complete. Construction of the lagging strand is somewhat more complex. The process of reading the lagging strand is discontinuous. And that's a lot of things that we don't probably need to get too much into, but the idea is when the newly constructed strand meets the prior segment, the primer is removed and the DNA ligase connects the two segments. These segments are called Okazaki fragments. So when the process is complete, the result is two identical DNA molecules, each one having the original strand and one replicated strand. This mode of synthesis is referred to as semi-conservative because the template strand is conserved from the original, now newly bonded to the synthesized strand. This slide here helps point out why proofreading is so important, and it's often mutations in the mismatch repair gene can increase our risk of cancer. So this process is done a gazillion times in our body, and any error can lead into mutation or a disease. So just to muddy the waters a little bit more, kind of go back a minute and let's watch this other part of osmosis to kind of put into words what I was trying to say about the transcription and all of this here. I think this is helpful because I know me reading this stuff and talking about it gets a little bit confusing.
Our DNA is like a library found in the nucleus of our cells with thousands of books. Some of these books, called genes, are extremely important because they carry the recipes for every single protein found in the cell. Some of these proteins are necessary in all cells, like cytoskeletal proteins, but other proteins, like neurotransmitters, are necessary only in certain cell types, like neurons. Gene regulation is what allows the right cells to make the right proteins at the right time. Now, on a molecular level, DNA is made up of two strands of nucleotides, so each gene is just a segment of this nucleotide sequence. And there are four types of nucleotides. These are adenine, guanine, thymine, and cytosine, or A, G, T, and C. Now, the entire DNA molecule is wrapped around structural proteins called histones that package the DNA into nucleosomes, like when we roll yarn into a ball to keep it compact and organised. Altogether, the entire yarn with the DNA plus the histones is called chromatin. The process of decoding the information stored in the DNA is called gene expression, and it includes transcription and translation. Transcription is where the enzyme RNA polymerase uses the gene as a template to create a molecule that can leave the nucleus. This molecule is called messenger RNA, or mRNA, and it has the same nucleotide sequence as the gene, with one tweak. It has uracil nucleotides, or U, instead of thymine. This message is encoded so that any three nucleotides equate a specific codon, which codes for an amino acid, or is a stop codon, which means that the protein is complete. In translation, specialized proteins in the cytoplasm, called ribosomes, use the mRNA template to create a string of amino acids that make up the protein. Gene regulation can occur at the level of transcription, post-transcriptional, which is between transcription and translation, or at translation. Let's start with transcriptional regulation. And to understand that, let's start with a deeper dive into transcription. Transcription begins with general transcription factors binding to a unique DNA sequence called the promoter, just upstream from the gene. General transcription factors help unwind the DNA helix so the RNA polymerase can transcribe the gene. However, general transcription factors can only bind to the promoter sequence if the DNA is not too tightly packed around the histones. Usually, genes that the cell uses frequently, like the hemoglobin genes in red blood cells, are packed more loosely and therefore easier to access. An interesting form of gene regulation, called epigenetics, can also make genes more or less accessible for gene transcription factors and RNA polymerase. Epigenetics refers to chemical modifications that selectively activate or silence certain genes without modifying the nucleotide sequence of the gene. These changes can occur in the histones or to the DNA itself. For histones, this usually happens through acetylation, which is adding an acetyl group to them, deacetylation, removing it, or through methylation, adding a methyl group. When an enzyme called acetylase adds an acetyl group to the histones, this decreases their attraction for DNA, so the genes become more easily accessible for RNA polymerase. On the other hand, if another enzyme called histone deacetylase removes an acetyl group, it has the opposite effect, increasing the attraction between DNA and histones, so DNA wraps more tightly around the histones and makes the genes less accessible. Methylation, on the other hand, can both increase and decrease transcription, depending on how many methyl groups are added. If only one methyl group is added, this decreases the attraction between the DNA and the histone, so the genes are easily accessible. If we add 2 or 3 methyl groups to it, however, the histone will repress or lock down, preventing the transcription of those genes. Epigenetic changes in DNA, on the other hand, are mostly based on methylation. The addition of a methyl group, usually to a cytosine residue, prevents general transcription factors from binding to the promoter, decreasing gene expression. As it turns out, DNA methylation also stimulates histone deacetylation, so that further inhibits transcription. So, let's say that based on the epigenetic modifications, our gene is exposed and ready to be transcribed. Sometimes cells need to increase or decrease transcription of a certain gene. And here's where the other types of transcription factors called activators or repressors get involved. Activators kickstart gene transcription through positive regulation. They bind to a DNA sequence called the enhancer, which is on the same DNA strand as the gene. Sometimes it's near the promoter, but other times it's further upstream or downstream. So, if the right stimulus, a ligand, reaches the cell, it gets the activator to move from the cytoplasm into the nucleus, and it binds to the enhancer. Once the activator is bound to the enhancer, it can loop the DNA around in such a way that it makes it even easier for general transcription factors and RNA polymerase to bind the promoter region. In addition, activators help recruit histone acetyltransferases, which also promote binding of RNA polymerase and transcription. On the other hand, there are repressors, which inhibit gene transcription through negative regulation. Repressors bind to a DNA sequence called the silencer, which is also on the same DNA strand as the gene. When the repressor is inactive, it's floating freely in the cytoplasm. But when the repressor protein is active, it moves into the nucleus, binds to the silencer sequence, and prevents RNA polymerase from binding the promoter, thus inhibiting transcription. Repressors can also recruit histone deacetylases, which also inhibit binding of RNA polymerase and transcription. The thyroid hormone receptor is an interesting example because it can act as a repressor and as an activator, depending on the situation. When there's no thyroid hormone in the cytoplasm, the thyroid receptor behaves like a repressor. It's bound to another protein called a co-repressor, and they bind to the silencer sequence of the DNA, and they recruit histone deacetylases, inhibiting transcription. However, once the thyroid hormone enters the cell, it binds to the thyroid receptor, and this induces a conformational change in the receptor. It's like the receptor has an epiphany, and now it behaves like an activator, releasing the co-repressor and binding a co-activator instead. The receptor and the co-activator then bind to the enhancer region, and they recruit histone acetylases, stimulating transcription. Once the gene is transcribed, we move into post-transcriptional regulation, which includes RNA splicing and processing. The freshly made RNA has the exact same nucleotide sequence as the gene. However, genes are made up of both coding sequences, which are called exons, and non-coding sequences, which are called introns. So RNA has to be spliced, meaning a molecular protein complex called a spliceosome comes in and it removes the introns from the RNA molecule, making messenger RNA, or mRNA, a much more efficient message. It's kind of like video editing, so you cut bits of the raw footage for a much cleaner final cut. Something interesting can happen here, which is called alternative splicing. For example, let's say there's an RNA strand like this, exon 1, intron 1, exon 2, intron 2, exon 3. Depending on which protein the cell needs to make, the spliceosome might do a few different things. It might result in 1, 2, or 3 of the exons as separate pieces of RNA, each going on to make a protein, or it could cut out the sequence from the start of intron 1 to the end of intron 2, thus taking exon 2 along. In this case, only 2 proteins will be synthesized, the products of exon 1 and exon 3. Ultimately, all of the post-transcriptional changes are made to the RNA. The resulting RNA sequence is now called messenger RNA, or mRNA for short. However, it's dangerous for mRNA to exit the nucleus like this, because lurking in the cytoplasm there are enzymes called exonucleases. Exonucleases really like cleaving nucleotides off the mRNA ends, and remember, they all code for amino acids now. So, mRNA undergoes processing, meaning it gets a cap and a nice poly-A tail. Capping basically means RNA gets a protective 7-methylguanine molecule at its 5' end. The poly-A tail at the 3' end is a long sequence of only adenine nucleotides, usually over 250 of them. In a nutshell, with a nice cap and a long poly-A tail, mRNA has a longer lifespan, so more proteins can be made using the same RNA molecule. Now, rarely, another form of post-transcriptional regulation can occur called RNA editing. This is where specific enzymes change the coding sequence by insertion, deletion, or substitution of nucleotides into the mRNA strand. These are called adenosine or cytosine deaminase acting on RNA enzymes, or ADAR and CDAR for short. ADAR converts the nucleotide adenosine to inosine, while CDAR converts the nucleotide cytosine to uracil. An example is apolipoprotein B, which is a protein that helps carry lipids in the blood. In the liver, the mRNA is fully translated into the protein ApoB100. In the intestine, the same mRNA undergoes a change. A CDAR changes the cytosine in the codon CAA, which codes for an amino acid, to a uracil, resulting in a UAA stop codon. That stops translation. As a result, the translated protein is a smaller variant called ApoB48. So, RNA editing helps determine where specific types of lipoprotein are synthesized. So, after splicing, processing, and occasionally editing, mRNA is finally safe in the cytoplasm, ready to be translated by a ribosome into a protein. The ribosome has two subunits called 40S and 60S, which work together to translate mRNA into a protein. Translation is divided into three main stages. There's initiation, which is where the 40S subunit of the ribosome recognizes the start codon and summons the 60S subunit to begin translating. There's elongation, which is where both subunits translate the codons into an amino acid sequence that makes up the primary structure of the protein. And finally, there's termination, which is where the ribosome recognizes a stop codon as a signal for 40S and 60S subunits to separate and release the protein. Translational regulation mainly happens during initiation. Normally, regulatory proteins called initiation factors have to bind to either the cap or the poly A tail before the 40S ribosomal unit can begin translation. They give the final go-ahead. Conditions like starvation or stress inactivate these initiation factors, which shuts down translation at this stage in order to save energy. Alright, so as a quick recap, gene regulation occurs at specific stages: transcriptional, post-transcriptional, and translational. Transcriptional regulation includes modification of DNA and regulation through specialized proteins called transcription factors. Post-transcriptional regulation includes RNA processing, which includes splicing and addition of a cap and a poly A tail, as well as RNA editing. Finally, translational regulation happens mainly when inactivation of initiation factors can prevent the ribosome from translating the mRNA, thus stopping protein synthesis.
Helpful. That was a lot of information, but again, we are not going to memorize all of those things, but I think it's very helpful to really have a nice detailed description of what happens during all of those stages. All right, so the next slide here talks about, again, we went from the significance of proofreading down into transcription. And let me find that spot. There we go. So in order for transcription to occur, they talked about this, we have to have a kind of a template that gives stop and starting instructions to the RNA polymerase that manages the process. So we have to have the promoter region, the coding region, and the terminator region. The promoting region instructs the RNA polymerase where to begin, as well as which strand is to be transcribed. The promoter region is not translated into part of the amino acid chain. The coding region is that portion of the gene that will give rise to the polypeptide chain and eventually the protein. And then that terminator region instructs the RNA polymerase where to stop, and this region is usually transcribed into the pre-RNA. So this here is transcription in a sense. So the RNA polymerase synthesizes a complementary RNA strand to a gene, a DNA segment coding for a protein. The result is a pre-mRNA molecule complementary to the length of the gene. So the process of transcribing the strand of RNA is similar to the replication of DNA. The strand of the DNA being copied is referred to as the template strand, and the other strand is the non-template strand, etc. So, once the entire strand of pre-RNA is transcribed, the molecule then must undergo post-transcriptional processing before it's ready to use, which is here. So while in the nucleus, the 5' cap and a poly A tail are added, and introns are removed, exons are spliced together to make the mature mRNA. I really like their cap and poly tail illustration in the osmosis video. It helps. I'm a visual learner as well, so it really helped me kind of understand better what they were trying to say. Again, it illustrates that better in my mind. So the pre-mRNA is shortened by spliceosomes that remove introns and polymerases that rejoin the exons that will ultimately be expressed. So we have a 5' cap and a poly A tail are added. We're just wanting to understand the general process here. And then in post-transcriptional processing that we're talking about now, we have to remember that the human genome contains somewhere between 20 and 25,000 genes. And one mechanism that makes it possible to do all of those is the alternative splicing of exons during the post-transcription processing. The poly A tail adds to the stability of the mRNA molecule, extending the time it takes before the molecule is degraded by the enzymes in the cytoplasm, so it makes it more stable. So what I said earlier is that one mechanism to make all of this possible is the alternative splicing of exons during the post-transcription processing. Multiple exons coded for by a gene will allow the different potential proteins to be translated in a mix and match fashion, and the mRNA then is made for the messenger RNA. So several possible proteins can be encoded by genes with multiple exons. It kind of makes it much more efficient, I would say that. Then we have the summary of transcription. So kind of what are we trying to say? The RNA polymerase initiates transcription at the start of the gene on the DNA, building a pre-mRNA from the template working in the 5' to 3' direction. Upon reaching the stop code, the RNA polymerase detaches and the pre-mRNA is processed by the addition of a 5' cap and a poly A tail. If the gene has exons and introns, spliceosomes made up of proteins and small nuclear RNAs remove the introns and join the exons together, which can complete the mature mRNA, making it more stable in the cytoplasm. The mRNA is translated to the cytoplasm to begin the process of translation. So, translation is the mechanism by which the sequence of the nucleotides in the mRNA is interpreted and a chain of amino acids is then produced. So we've transcribed it, we have the recipe, and now we have to make the recipe. So during this phase of the protein synthesis, 2 additional types of RNA are introduced to the process. Ribosomes are factories made of ribosomal RNA, which is rRNA, and the proteins. So these complexes read the mRNA message and then synthesize the protein chains based on that message. The transfer RNA carries the amino acids one by one to the ribosome, where then each amino acid is added to the growing polypeptide chain. In order for the process to work, though, the tRNA must have a mechanism for recognizing which amino acid is to be delivered to the ribosome. And the ribosome must have some way of knowing when to end and when to begin. So this is the example on your screen here of the translation. Remember, translation occurs in the cytoplasm when the small and large ribosomal subunits attach to that messenger RNA, and then the ribosome moves along the messenger RNA with the tRNAs binding briefly to bring individual amino acids to be added to this growing chain, and then they exit. One more part about translation here. We have post-translational processing. So the initial product undergoes additional modifications before the functional protein has reached its final form. So the post-translational processing can include folding of that polypeptide chain into the three-dimensional shape that we talked about in chapter 2. So the protein folding. The chemical and biochemical foundations combine the emerging protein with the other polypeptides to create that quaternary structure, and then sometimes remove some of the amino acids from that chain. So the folding process may occur either during or after translation, and it's often facilitated by chaperone proteins. So phosphate, carboxyl, methyl, and carbohydrate groups may be added to alter the function of the resulting protein. So some proteins include a sequence of amino acids called a signal sequence that is used to direct the protein to a particular cellular compartment. So in other words, we have the ribosome function in translation can also do some things in the post-translational process. This is the transfer RNA molecules that have a looped structure that help do the transferring of the RNA. At the end of the loop is the anticodon destined to hold its complementary site on the mRNA. After losing their amino acids, the tRNAs leave the ribosome and await attachment for the next molecule. To say that. All right, now we're going to go on to a new thread of thought here in chapter 3, which is the concepts in genetics and genomics. This is an example of genetic coding. Gotta love that, where the stop codes are, etc. So with 3 bases in each codon and 4 choices of base, there are 64 possible combinations available to code for the 20 amino acids, 1 start codon and 3 stop codons. Looking at that table, note that all of the amino acids except methionine and tryptophan have more than 1 associated codon. Often the codons for an amino acid are only different in 1 position, so this reduces the possibility that a change of a single base or a mutation will change the whole sequence of the protein. All right, more into the chromosome terminology. The 2 copies of each chromosome are termed homologous, I can never say that word, chromosomes. And an individual's genome is made up of 2 homologous copies of each autosome, and then we have the 2 sex chromosomes. Within each of the chromosome pairs, 1 chromosome was contributed by the egg and 1 was contributed by the sperm. These are referred to as the maternal and paternal chromosomes. Each chromosome has a centromere, a region that will become the connection point between sister chromatids during mitosis. And remember, the centromere is the point of attachment where during mitosis those spindle fibers attach here to pull them apart into 2 new cells. Some more gene terminology that I wanted to go through too here. I'm sorry, this is, the gene mapping. Yep. Okay, gene terminology is in your book as well. The gene locus or loci is indicated by a shorthand nomenclature that includes the chromosome, the arm of the chromosome, and the gene's location within that chromosome. So we have a locus, allele, genotype, phenotype, homozygous, which means having identical alleles, which is the version of the gene, on both the homologous chromosomes. Heterozygous is having different versions of the gene which is a variation in the sequence of the gene on the maternal and paternal chromosomes. And then we have a wild type and then mutations. All right, genetic variability. The 3 billion base pairs of the human genome have undergone tremendous changes in the course of evolution, and the genome alterations and variations account for many aspects of evolution. Some of the common types of alterations affect the genes themselves. That's about 1% of the genome. But the other alterations affecting the non-coding regions that serve other functions. Not all functions of the non-coding regions are currently known. So some of the common alterations that we do know can be a single nucleotide polymorphism, a gene duplication, or variable number tandem repeats. The term genetic mutation has sometimes been used to describe all the changes in DNA, but the term genetic variation is also coming into use as a way of describing the vast and subtle variability that can make up the genome. For the purpose of the book, they focus on genetic mutations that are known to cause disorders, while acknowledging that the most common human disorders or diseases are complex and polygenic in origin. But genetic mutations can occur spontaneously, but they can also arise as the consequence of chemical or radiation-induced damage. The damage may be in the form of reactive oxygen species, chemical mutagens from the environment or occupational exposures, radiation even in the sun, affecting the skin, or failure of cell protective strategies. Mutations may impact the final protein product in a quantitative or qualitative manner. So these are some of the point mutations that can affect the coding regions. They can be silent, missense, or nonsense. So silent point mutations is a change in the genotype but does not change the phenotype. Missense is altering a nucleotide changes one amino acid within the whole protein product. And then nonsense alters a nucleotide, inserts a premature stop codon. The protein is truncated and non-functional. That's just one of their examples. Here's some of the point mutations, etc., and what that looks like. We can also have insertions and deletions. When you have a removal or an addition of the extra base pairs, that will change the actual number of base pairs in a coding or non-coding sequence. If it occurs in a coding region, it can affect the amino acid sequence. This is such kind of what happens when people develop cystic fibrosis, for example. Can be a deletion or insertion mutation. Then they talk about frame shift mutations, and that's kind of what they show on the right side there. The deletion or insertion of one or more bases not divisible by 3 in a coding DNA sequence changes the reading frame for transcription and produces a frame shift mutation. Okay. So if you screw up in one of the sets of 3, then you shift everything, and now you have a major problem. I know we went over this all pretty quickly, but it's kind of just straightforward reviewing of terminology that can then lead us into some of the other diseases we're going to talk about. The trinucleotide repeats also you can read here. When you repeat something, it can lengthen the gene and have a variable impact on the product of the individual. So it can be known as expansion. All right, so some of the diseases that we talk about for this chapter. First one is Huntington's disease. Huntington's is an adult onset fatal neurodegenerative disease, and it's caused by excess trinucleotide repeats in the gene coding for the Huntington. So individuals with Huntington's disease have over 35 to 40 CAG repeats, when you're supposed to really only have a normal number of 17 to 20. So because the CAG codes for glutamine, the translated protein has a longer stretch of glutamines than normal. So over time, the mutant Huntington protein accumulates and it damages the neurons and it causes neurodegeneration. Huntington disease manifests in individuals around the age of 35 to 45 and is characterized by uncontrollable chorea, which is brief, twitching, non-purposeful movements that occur sporadically, which progresses to greater motor dysfunction as well as cognitive loss and psychiatric manifestations. It's a devastating disease. What the book is now doing is it's trying to introduce diseases to kind of tie the topics of the general gene problems that we're talking about into an example of a disease that we can kind of correlate together. Huntington disease and the other trinucleotide repeat diseases exhibit genetic anticipation. So each generation tends to have a worse phenotype and earlier presentation. Some of the other trinucleotide repeat disorders include Fragile X syndrome, myotonic dystrophy, spinocerebellar ataxia, and Friedreich ataxia. This gives an example of what that looks like for Huntington's. All right, let's review some of the Mendelian genetics and Punnett squares. And again, we have a great option here in osmosis to give us a nice refresher and explanation of what that means.
Genetics is the science that studies inheritance, or the way parents transmit certain traits to their descendants. And Mendelian genetics refers to Gregor Mendel, an Austrian monk, who studied inheritance by experimenting on pea plants. He cross-pollinated the flowers of different plants together, took the seeds that developed from the pairing, planted those seeds, and took careful notes on the types of peas that resulted in the subsequent generations. You might say, as a monk, he was trying to find his inner peas. Now, in addition to having lots and lots of peas in his garden, he helped to formulate two important laws, the law of segregation and the law of independent assortment. So to start out, Mendel took plants with violet flowers and plants with white flowers, and crossbred them. This original group of flowers are called the pea generation, as in parent. And then when he obtained some peas, he planted them and got more plants, and the flowers in this offspring generation were called F1, or filial 1. It turns out that the F1 generation consisted of all violet flowers, so we called the violet trait dominant, while the white trait, which appeared to be lost in the F1 generation, was called recessive. Next, Mendel let the violet flowers in the F1 generation cross-pollinate amongst themselves, and when they formed peas, he planted them again. From this, he got more plants, and the flowers from the second generation of plants he called filial 2, or F2. It turned out that some of the plants in this F2 generation had white flowers, whereas other plants had purple flowers. In fact, the ratio is about 3 violet flowering plants for every 1 white flowering plant. Based on this experiment, Mendel drew a few conclusions. First, since the F1 violet flowers had some offspring plants that produced violet flowers and other offspring plants that produced white flowers, it meant that the F1 plants must have contained both of these elements. The inheritable elements of pea plants are its gametes. So that meant that the gametes of the F1 plant contained either the dominant violet trait or the recessive white trait. The F2 plants are created with one gamete from each parent. And Mendel worked out that the white flowering plants resulted when they received both white flower elements, and that plants that had at least one violet flower element from either parent would produce violet flowers. Mendel didn't know this at the time, but the element he was referring to were segments of DNA called genes that encoded each flower color. These genes were located on specific parts of chromosomes, called loci. Different versions of a gene are called alleles. And in the case of the flowers, there were 2 alleles, a white and violet allele for flower color. A helpful way to visualize Mendel's experiment is to use a Punnett square. Imagine a box with 4 squares in it, where we put the genetic information of one parent, or genotype, on the horizontal row and the other parent on the vertical column. The dominant allele, represented with a capital letter, codes for a violet flower, and the recessive allele, represented with a lowercase letter, codes for a white flower. The letter we choose doesn't matter, so let's use capital P for the violet flower allele, and a lowercase p for the white flower allele. In the P generation, Mendel used pure breeding plants, so their genotype was 2 of the same alleles. In other words, both of the parent plants in this generation were homozygous for flower color trait. Homo meaning same, and zygous referring to the male and female alleles. The violet pea plant had 2 of the same dominant alleles, capital P, and therefore had all violet flowers, whereas the white pea plant had 2 of the same recessive alleles, lowercase p, and therefore had all white flowers. Now, the observable trait that results from the genotype is called the phenotype. In this case, the phenotype is the flower color. So when the violet and white flowering plants were crossbred, each offspring got a dominant allele from the violet flower parent and a recessive allele from the white flower parent. Since the 2 alleles are different, these plants are all heterozygous, meaning that they have hetero or different alleles for the flower color trait. The phenotype of these heterozygous plants was that they all had violet flowers, because the dominant capital P allele masks the recessive lowercase p allele. Now when we breed any 2 of these heterozygous plants in the F1 generation, we can make a new Punnett square, with the capital P lowercase p genotype of one parent on the horizontal row, and the same capital P lowercase p genotype of the other parent in the vertical column. When we use the Punnett square, we get 1 offspring with a capital P genotype, 2 with the capital P lowercase p genotype, and 1 with the lowercase p genotype. The 3 plants with at least 1 capital P allele will have a violet flower phenotype, and the 1 plant with a homozygous lowercase p genotype will have a white flower phenotype. This was the ratio of plants that Mendel observed in the F2 generation, and it helped establish the law of segregation, which states that alleles segregate, and that offspring acquire 1 allele from each parent. Now, it turns out that in addition to flower color, Mendel also observed the seeds of his pea plant, specifically their color and texture. He noted whether the seeds were yellow, which we'll call the dominant big Y allele, or green, the recessive little y allele, and whether the seeds were round, which we'll call the dominant big R allele, or wrinkly, the recessive little r allele. As before, Mendel started with pure breeding plants. One of them was homozygous dominant for both traits, which means that it was capital Y genotype for the color trait, and capital R for the seed texture trait. So this plant's phenotype was that it had yellow, round seeds. The other plant was homozygous recessive for both traits, which means that it had little y genotype for color trait, and little r genotype for texture trait. So its phenotype was that it had green, wrinkled seeds. So Mendel cross-pollinated these 2 plants, and the result was that all of the plants in the F1 generation got capital Y, capital R from one parent, and lowercase y, lowercase r from the other parent, and therefore were capital Y, capital R, lowercase r. So far, so good. But then Mendel bred 2 of these F1 plants with one another, and things got interesting. Let's put this in a Punnett square. For these 2 traits, there are 4 different combinations for each parent. Capital Y, capital R, capital Y, little r, capital R, little y, and little r, little y. When we crossbreed the plants, we can expect that the F2 generation will have seeds that have 4 different types of phenotypes. 9 are yellow and round. These have at least 1 dominant capital Y and 1 dominant capital R. 3 are yellow and wrinkled, those that have at least 1 dominant capital Y and 2 little r's. 3 are green and round, those that have 2 little y's and at least 1 dominant big R, and 1 that's green and wrinkled, the one that has 2 little y's and 2 little r's. And that's what Mendel got, a 9:3 to 3:1 ratio. And this helped establish the law of independent assortment. that the genes for seed color and seed texture were assorting independently of each other, and they weren't somehow influencing one another. In other words, having one trait did not make having another trait any more or less likely. This law is generally true, except in certain situations, like when two genes are located really close to each other on a chromosome. When that happens, it's called genetic linkage, and the two genes start to move together more often than not, and therefore don't assort independently. Alright, as a quick recap. The law of segregation states that inherited alleles are separated when producing gametes. And the law of independent assortment states that the alleles get distributed to offspring randomly, and without regard to what other allele the offspring might have received.
That osmosis video is going to explain it probably better than I'm going to. So I'm glad we had that chance to watch that. It explains it very well. The other thing to remember is that the family history can provide clues of genetic nature of diseases, but there are genetic disorders that can occur from like spontaneous mutations, right? So that we know that some things are genetic, but sometimes we can also get diseases just from spontaneous mutations, which is kind of what we're also needing to remember when we're talking about pathology for humans. This here is roughly what we just saw in the osmosis video. It talks about autosomal dominant. Males and females are roughly equally affected. Then we have autosomal recessive. The heterozygous carriers do not express the phenotype. Then we have that Punnett square estimate, right? So if you had a non-affected parent had the G and the G, so they had two dominant alleles, but the affected parent had the recessive allele. If they had children together, they'd have a 50% chance of the child being affected. Here's some of the autosomal recessive patterns to look at. Really just to be familiar with what we're talking about when people ask us about genetic diseases. We have some of these X-linked inheritance issues too. Several factors will influence the phenotypic presentation with genotypes that have disease-related genes. So penetrance and expressivity. So penetrance describes the likelihood that the individual with the genotype associated with the disease actually will present with the disease. So some disorders are 100% penetrant. Sickle cell and Huntington's, there's no chance that you're not going to actually express the disease. The penetration of that disease, so to speak, to come through is 100%. But some of them, other diseases we have a variable penetrance. So even though you may have it, you may not actually present or express show the disease, such as like hemochromatosis. Then expressivity describes the variability of phenotype among those with a disease-related genotype. So the presentation can be mild, moderate, or severe, but if the disorder is 100% penetrant, all those with the mutated gene have the disease to some degree. So the classic one is cystic fibrosis or osteogenesis imperfecta. But cystic fibrosis, you can have people that have a very mild disease or people that have a very severe presentation of their disease. Marfan syndrome. Okay, these are examples now that the book wants us to kind of relate to. So Marfan has an autosomal dominant inheritance pattern, but 25 cases are new mutations. So affected male or female child can be born to unaffected parents. So yes, it's typically genetic, but also can be just a spontaneous mutation. So the penetrance is 100%, but the expressivity, mild, moderate, severe, is variable. It results in abnormalities in the connective tissue formation and can result in a tall stature, hypermobile joint scoliosis, aortic dilation, and dissection, and then mitral valve prolapse. So when we think about Marfan, yes, it can be an autosomal dominant inheritance genetic disease, but it can be a new mutation as well. And then BRCA1 and BRCA2 are hereditary for breast and ovarian cancer. And although most cases of breast cancer are caused by new mutations, there are BRCA1 and 2 mutations that do increase risk. So if you are a carrier of BRCA1, you are more likely to get breast or ovarian cancer. Definitive identification of the relevant gene products and roles are not currently available, but we'll talk more about this in Chapter 7. Having one abnormal gene is not sufficient. So even if you're a BRCA carrier, it's not 100% that you are going to get that kind of cancer. You need a second hit down the road to also activate that. Remember, we'll talk about this further, but BRCA1 and BRCA2 can increase the woman's chance of developing breast and ovarian cancer, but it also increased the risks for the man to develop male breast cancer and prostate cancer. Okay, coronary artery disease we know is genetic as well, can be. It's multigenic, multifactorial. Family history is considered a risk factor. However, it's not 100%. Certain rare single gene disorders will definitely increase your risk and lower the age of onset. So if you have familial hypercholesterolemia, so it's passed in the genes of the family. I worked with a physician who was 38 and was on a slew of cholesterol medications, had very difficult time managing his cholesterol, and he was a marathon runner and in the best shape of his life, but he had familial hypercholesterolemia. Another one is familial defective apolipoprotein A, another condition. We also know that there's other gene mutations that increase your risk for cardiovascular disease. This SLC01B1 increases myopathy risk from statins, and the CYP2C19, certain mutations prevent the hepatic activation of the clopidogrel prodrug, so you don't respond to Plavix. Depression. We have relative and twin studies that do support a genetic component, but the gene has not been identified. Therapeutic efficacy of the antidepressant drugs varies widely, and we now know that we can do genetic testing on people to see if they carry the genes and which of the medications they're going to likely metabolize the best and therefore should work the best for them. So a family history or a pedigree determination is the first step. The family genes mnemonic is groups early neurodevelopmental, exceptional, and surprising. Later, when we get into advanced physical assessment, you're going to need to do a genogram and practice this as well. We can do karyotyping. We're not going to get into a lot of detail about that right now. We now know that in a lot of cancers, for example, we can do medications that can work with antibiotics or psychopharmacology, etc., to treat. We can have laboratory manufactured DNA strands synthesized. Each probe is spotted then on the chip, and the patient DNA is isolated, purified, and cut into short strands. We have a lot of different hybridization kind of going on in the world of DNA. So this is an example. So they identify transcripts that are increased in tumor cells that could be driving the tumor growth. So you can see what a normal cell would look like versus the cancer cell. Other transcripts are decreased in tumor cells. Some transcripts are unchanged between normal and tumor cells, not likely to be involved in tumor growth. So they put them through the cell culture, the RNA isolation, the reverse transcription and labeling, and then hybridize the cDNA probes to oligosequences on the microarray, and then we get our results there. RNA technologies, we're learning a lot more about as well. And so we have some of these RNA therapeutics that can target specificity predictability and cost effectiveness and safety. So we can have gene editing to help treat us in cancers is kind of what we're working towards in the future. Okay, I do want to talk more about the mRNA vaccines and microRNA. So kind of skipped forward a little bit to get to some of these things. I do want to talk a little bit more. Okay. Popular interest in the vaccines developed using mRNA certainly peaked during COVID-19. But the history of the mRNA vaccines, the first human trial of this mRNA vaccine, of any mRNA vaccine, was developed in 2008. And we've started on that technology in the 1970s. So upon injection, these vaccines introduce an mRNA transcript, which is transported into the recipient cells. Once within the cell, the mRNA transcript enters a ribosome and is translated as any other mRNA, resulting in the production of a polypeptide chain. Okay, so the COVID-19 mRNA vaccines code for the spike protein that develops on the surface of infected cells. In response to the presence of this foreign protein, our normal immune system processes create both antibodies and memory cells specific to the virus. So the vaccines have several advantages over the other types. mRNA vaccines do not rely on the introduction of either live or attenuated viruses to create their immune response. So they say vaccine production does not depend on the creation of large amounts of the virus. Additionally, mRNA transcript can be produced relatively easily and in large quantities. Also, while popular concern was expressed about the ability of mRNA vaccines to alter the host's DNA, the structure of mRNA does not permit the molecules to enter the cell nucleus and contact the host DNA. So, in theory, they're supposed to be very safe. Then we have microRNA. Is a small fragment that regulates gene expression by downregulating target genes. MicroRNA are evolutionarily, oh my gosh, you guys, evolutionarily conserved and involved in various metabolic processes, making them potential biomarkers for disease diagnosis. So those are being studied, and you'll probably see in the future as well. We have some of these other approaches and things happening in genetics, and you guys are reading those in your book. There's nothing really to talk about. We have the direct-to-consumer testing. There's some interest in that. It may fall to the clinician, though, you guys, to explain the difference between correlation and causation, odds ratio, risk prediction, and to allay patient concerns. All right. Epigenetics is, okay, this is the term that has undergone several changes and meanings, but essentially means the study of heritable changes that do not involve alterations in DNA sequencing. Okay, let's listen to the little blurb on osmosis about epigenetics.
Epigenetics is a process of gene regulation, or turning genes on and off. Think about it. You have about 37 trillion cells, and over 200 different types of cells in your body. For example, there are muscle cells for looking great on the beach, as well as neurons that tell your muscles to flex when it's time to show off. And both muscle cells and neurons have the same origin and genetic material, meaning 46 chromosomes, with each chromosome made up of a single DNA molecule. Along that chromosome are sequences of DNA that code for genes, with thousands of genes on each one. It makes sense that there would have to be a process to control all of those genes. Now, it turns out that DNA is a very long molecule, over 2 meters when it's fully stretched. So to save space, DNA is wrapped around special proteins called histones. Now, histones actually come in groups of 8, 4 stacks of 2, kind of like poker chips. And the DNA molecule wraps around each group of 8 histones twice, forming a nucleosome. Different sections of DNA, meaning different genes, wrap around different stacks of histones. Finally, the nucleosomes are packed together even more tightly, resulting in chromatin, which looks like threads of cotton candy within the nucleus. Now, a cell type boils down to what a cell does, and in turn, what a cell type does boils down to the kinds of proteins it makes to carry out its role. Proteins are made based on genes, so our collection of genes, or genotype, is actually like an incredible wardrobe. It contains something for every occasion. And different cell types wear different attires. For example, our muscle cells are usually doing the hard work of contracting and relaxing all day, so they'd require the equivalent of athletic gear to do their job. Posh neurons, on the other hand, might prefer a tuxedo to tend to their synapses. So the muscle cell needs only certain parts of that wardrobe, and the neuron needs a very different part of that wardrobe. This is achieved through selectively activating or silencing certain genes. The final appearance of how a cell looks depends on which genes are activated, and we call that the phenotype. All of this happens through epigenetics, which specifically refers to mechanisms that can selectively activate or silence certain genes without modifying the nucleotide sequence of the gene. Let's start with histones. Histones can be influenced to either release their DNA or lock down their DNA through chemical changes, like acetylation or methylation. For example, when an acetyl group is added to the histone, there's less attraction between DNA and histones. As a result, the genes are more exposed and therefore more easily transcribed. Removing the acetyl group through deacetylation has the opposite effect. DNA wraps tighter around the histones, so the genes are less easily transcribed. Methylation, on the other hand, can both increase and decrease transcription, depending on how many methyl groups are added. If only one methyl group is added to the histone, this decreases the attraction between the DNA and the histone, so the genes are easily accessible. But interestingly, if 2 or 3 methyl groups are added to the histone will repress or lock down the DNA, preventing the transcription of those genes. Epigenetics can also include a direct modification of the DNA in a way that doesn't change its nucleotide order. Genes are just a particular sequence of the 4 nucleotides: adenine or A, thymine or T, guanine or G, and cytosine or C. These 4 bind to each other specifically. A on one strand binds to the T on the other, while C pairs with G, and vice versa. Epigenetic modifications usually happen in regions of DNA that have a lot of cytosine and guanine nucleotides one after the other, and these regions are called CpG sites. In these regions, the cytosine residues can undergo methylation, so a methyl group is now hanging off the cytosine, but the sequence of nucleotides remains unchanged. A lot of DNA methylation can silence gene expression, so if a gene contains many methylated cytosines, it'll be less likely to get expressed. You can think of methylated cytosines in DNA as thorns on a rose stalk, making that rose much less likely to be picked. It's also important to remember that rather being an on-off switch, these epigenetic modifications just help to tune the frequency of something happening, like a dimmer switch. So if someone is truly after that rose, it might still get picked, but there'll be some pain involved. Now, the key is that epigenetic modifications on both histones and DNA can happen throughout a person's lifetime, from fetal development through the aging process. And various factors like recreational drugs, medications, diet, and exercise can all play a role. Many epigenetic changes are reversible, so changing these factors can affect these epigenetic modifications and gene expression. In fact, epigenetics helps explain why identical twins have the same DNA but often have subtle differences in their appearance, their personality, and even in the diseases that they might develop. Epigenetic changes are also potentially inheritable, but how chemical modifications of DNA get inherited is still an area of intense research. Alright, as a quick recap, epigenetics refers to the chemical modifications that can selectively activate or silence certain genes without modifying the nucleotide sequence of the gene. For histones, this can happen through acetylation, which activates gene expression, deacetylation, which deactivates gene expression, or methylation, which can do both. Finally, DNA can be epigenetically altered by methylation of a cytosine residue in a CpG site, which silences gene expression.
Again, the osmosis videos explained this much better than I would, understanding that we can have methylation and acetylation that can make the genes more accessible, and then we can have deacetylation that will decrease the likelihood of the transcription. Moving on to prenatal and neonatal screening. This is important for us because you are going to be taking care of pregnant adults. The PKU we talked about last time, we need early intervention with a diet to detect it, and if it is found, then we have to have these pretty extreme diet changes. We have other disorders that are diagnosed before birth, maternal alpha-fetoprotein, Down syndrome, trisomy 18, Turner syndrome, etc. Newborn screening panels, again, we do need to understand what they are and to be aware of them in all states. We have a standard panel of tests that's recommended within a few days of birth. The newborn screening includes the most common disorders as well as many inborn errors of metabolism and endocrine conditions. Early genetic testing is becoming much more common for infants with a variety of problems identified soon after birth. All right, you stuck with me. We made it through in an hour and a half of Chapter 3. Chapter 4 coming up is also going to be an intense technical couple of chapters, and then after that we get into Chapter 5 through the rest, and that's a lot more into actual disease states and body systems to talk about physiologic changes that way.